For this video, I'm going to be discussing the major pathways of complement activation. The first pathway is referred to as the classical pathway. This pathway is initiated by the binding of an antigen to an antibody in an antigen-antibody complex, and it is considered to be part of the adaptive immune response. Not all antibodies are able to activate this pathway, only some subclasses of IgG and some complexes of IgM are capable of it. When the antigen and antibody bond, it induces a conformational change in the shape in the Fc region or the non-antigen binding region of the antibody. When this happens, it exposes non-antigen bound IgM circulating in the blood is incapable of bonding to the C1 complex because its configuration is flat and there are no C1q binding sites exposed. However, when pentameric IgM is bound to an antigen, it undergoes a conformational change into what is called the staple configuration. In this configuration, there are at least 3 C1q binding sites exposed. Circulating non-antigen bound IgG also is incapable of bonding to the C1 complex. Monomeric IgG, subclass IgG1, is capable of binding to the C1 complex. However, it only has one binding site per molecule. So this means that several IgG molecules are needed to activate the classical pathway, whereas only a single IgM molecule is needed to initiate it. Upon the bonding of C1q to the CH2 domains on the Fc regions of the antigen-bound antibody, one of the C1R molecules changes shape and turns into an active serine protease enzyme, which then cleaves and activates the second C1R molecule. The two C1R molecules together then cleave and activate the two C1S molecules. C4b, which is a substrate of C1s, binds to the surface of the target membrane. C1s is divided into two substrates, C4 and C2. C1s then cleaves C2, leaving the substrates C2 and Ca. The second pathway of complement activation is the lectin pathway. This pathway is initiated when soluble lectin complement proteins recognize an antigen. The lectin pathway is a part of the innate immune system. It is similar to the classical pathway in that it uses the same C3 convertase. It uses C4b2a. This pathway uses lectins instead of relying on antibodies to recognize the threat. Lectins are proteins that recognize specific carbohydrate components found on the surface of microbial cells. Mannose-binding lectin, abbreviated MBL, was the first lectin found to be capable of initiating complement cascade. MBL in the serum is often associated with MASP proteins. MASP stands for MBL-associated serine proteases. MASP2, one of the MASP proteins, can cleave C2 and C4, which produces C3 convertase in essentially the same way as the classical pathway. The lectin pathway differs from the classical pathway in that it replaces the C1 complex with the MASP proteins to cleave and activate the C3 convertase to form the C5 convertase, which is also the same as the classical pathway. It is C4b2a3b. The third major pathway of complement activation is the alternative pathway. This pathway evolved as a way to protect the host from invaders that evolved to become resistant to the classical pathway. It is also considered a part of the innate immune system. It uses its own set of C3 and C5 convertases. The C3 convertase is composed of one molecule of C3b and one molecule of big B little b, which forms C3b big B little b. C5 is formed by adding the second C3b to make C3b big B little b C3b. The alternative pathway can be activated in 3 different ways. The first way is the alternative tick-over pathway. The second pathway is the alternative prepared and activated pathway. And the third way is the alternative protease activated pathway. The alternative tick-over pathway is initiated when C3 undergoes spontaneous hydrolysis. The product of this reaction is C3H2O. C3H2O binds to factor B in the presence of magnesium ions. Factor B is cleaved by factor D, leaving substrate Ba and Bb. Ba diffuses away, and Bb binds to C3H2O. C3H2O in the plasma in its liquid state has convertase activity. It cleaves many C3 molecules into C3A and C3B, acting as the C3 convertase. C3B molecules will then bind to microbial surfaces if there is an infection present. Factor B binds to C3B molecules. Factor D cleaves factor B again to form C3B, big B, little b complexes. These membrane-bound complexes also have C3 convertase activity, and this will actually take over as the primary C3 convertase in this alternative tick-over pathway. C3b, big B, little b binds to properdin for stabilization. The bonding of these two molecules generates large amounts of C3b, which will bind to more factor B. C3b binds to C3 convertase, C3b, big B, little b, to form the C5 convertase, which is C3b, big B, little b, C3b, which forms the membrane attack complex, or the MAC. In the alternative tick-over pathway, properdin serves as a regulatory molecule that stabilizes the C3 convertase. However, in the alternative properdin activated pathway, properdin has been shown to act as a pattern recognition receptor, which is abbreviated PRR. It recognizes patterns and activates the alternative pathway. This pathway relies on lower levels of C3b, and it can actually provide greater selectivity than the tick-over pathway. The third and final alternative pathway is the alternative protease activated pathway. This pathway uses protease cleavage and conformational alterations, so changes in the shape of the proteins to modify their enzymatic activities. This amplifies various steps of the pathway and produces a lot of substrates through this. Thrombin is used to cleave the C5 in this pathway, so it acts as the C5 convertase in this case. The three complement pathways come together at the formation of a C5 convertase, whichever way that it is produced. For the classical pathway and the lectin pathway, it is the C4b to A3b, and for the alternative pathway, it is the C3b, big B, little b, C3b, except in the case of the thrombin-initiated pathway, which was the last one that I talked about. The result of the C5 convertase activity is all the same, and so after this, the complement cascade goes on to cleave the C5s, and the C5b is generated on the target cell surface, and it will provide the binding site for the MAC components.